Hi everyone, welcome to our first lesson of our second unit, which is genetics. Today we're talking about sort of like an introduction to DNA, bit of an introduction to what is heredity. And really in this unit, we're going to be exploring how traits get passed from parents to offspring and how they move through generations, as well as why we see the physical aspects that we see. Why do we see that some traits are covered up and others are shown more prominently? So that's like a little bit of an intro what we're doing this unit. There are going to be some things which we just touch upon this year, and then in grade 12 you go into far more detail with. So I'll make mention of a few of those things in this lesson as well. So I wanted to just start off by this question, and hopefully you were able to think about this a little bit, even in grade 10 science. Why do cells divide? So there's a few different reasons as to why. One is for reproduction. One is to repair or regenerate. So if you have a damaged tissue as well, which I mentioned here, you want to replace it. Sometimes you need to actually do it for growth as well. So that's sort of like this regenerate part is if the organism needs to grow, the cell doesn't just get super large, it just starts to divide. So cell division we focus on in grade 10. You will tomorrow, or excuse me, for lesson 2, you will do a small activity, which is really just reviewing mitosis. So that is what to look forward to there. I just want to mention heredity now. So heredity is essentially how things are passed, as I mentioned before, from parents to offspring. And genetics as a general topic is how that happens, okay? And I mentioned right here, how does DNA instruct cell function? That has to do with the code of life, and that's really what we're focusing on in this unit. Next year in grade 12, you will do a whole unit which is called molecular genetics, which is composed of a few things. Number one is structure of DNA. How does the DNA get copied? Because right now, and in grade 10, all we've said is DNA is replicated, but there's actually a very sort of long process, which involves lots of different things, which we'll talk about in grade 12. So it's not as simple as just it copies the DNA. It has to do so really perfectly as well. So that is something to look forward to for grade 12. So for genetic material, for humans, it's deoxyribonucleic acid, or DNA for short. This refers to the chemical structure, and I've mentioned a couple of times before. In grade 12, it's really the chemistry of life, biochemistry. So you will learn further about that structure next year. I'm going to just show really the basics of it today. Most human cells have 46 chromosomes. Now, in lesson 4, we're going to talk about some people have an atypical number, which means they have more or less than 46. That can happen. And those typically will result in a genetic condition for the person that has that. But humans typically have 46, and those come from the egg and sperm cell of our parents. So the egg and sperm cell of our parents, they have half as many, what we call a haploid number. They have 23 each. So a little bit about structure of DNA and more like definitions of DNA. We have shown here is just a single line. We call that one chromosome, which is unduplicated. Sometimes you'll hear me call it just a single chromosome. The one over here, which looks like an X shape that we often draw, that is a duplicated chromosome. So this guy over here is after cell division has happened, after DNA synthesis has happened. So this has already been copied. Now, if everything has gone to plan, these two sides of the X should be identical, okay? So this we call a duplicated chromosome. And I want to mention something that's going to come up in some videos we watch later. There's also the centromere. And the centromere, you might remember from mitosis, that is a connection point for our spindle fibers to help us move around the chromosomes during cell division. When we're counting chromosomes, we are always counting the centromeres. So this is a single chromosome. This is also a single chromosome because in both cases, there's just one centromere. Now, when we get into meiosis in lesson 3, we're going to discuss that we will start calling this a single unduplicated chromosome, or here a duplicated chromosome. But either way, it's still one chromosome. The other new term for some of us is down here, sister chromatid. So I mentioned about this X shape. It's really just one single chromosome that's being copied into two, which we're seeing as the X. So when we talk about the individual sides of this, we can call each of them the sister chromatid, which just means they are exact copies of one another. So a bit more on that in lesson 3. Just to mention here, there's also labeled as genes. So genes are specific sequences of DNA which code for some specific trait or some specific protein. We're not going to look at this year in details how that gene becomes that instruction. That is a focus of grade 12. But we will talk about genes for sure. We just won't see the actual process of how they become the trait itself until next year. So we also describe this as a locus. A locus just means a location somewhere on the chromosome. So we'll find out later in the unit that the human genome has been mapped out. So we do know the location of many of the genes that relate to humans. There are between 20 and 25,000 different genes in our genetic set. So those 46 chromosomes have lots of genes on them. So we don't know the function of all of those yet. That is what geneticists are working on. Okay. We're going to move into now the structure of DNA. Again, I mentioned biochemistry next year, grade 12. You will spend a lot more time talking about this, so this is really just an introduction. If you're curious to read some more, please feel free to check that out in the textbook or just Google it. You can find lots of information. But I wanted to just show the basics of it here, which is how was it discovered. So someone called Rosalind Franklin, she's a famous scientist. She found out in 1952, she took this very famous photograph of DNA. And what it is essentially doing is like looking at a side view of a DNA helix. So think of the helix as a spiral, and you're essentially looking down the middle of the spiral. So that's what this picture is taken of. Now, these two people, Watson and Crick, they're actually sort of famous for, I'll say, is actually taking her picture and then sort of devising what the actual chemical structure is. So in the 1950s, I'll just mention, women were not very well respected in science. And so, unfortunately, Rosalind Franklin did not get the credit that she really deserved for figuring this out. And in fact, she ended up dying because this picture uses X-rays to take it. And as we know now, X-rays are carcinogenic. And so unknowingly, she was actually getting some DNA damage and she developed cancer, which she eventually died from. So where I'm going with this is these two, Watson and Crick, they essentially took her work and they sort of like made it their own. So they basically developed what we now use as the DNA helix. So I want to talk a little bit about organization now. So DNA is essentially what we call polymer, which is a long strand of things that repeat. And the monomer, which is one single piece in DNA, is called a nucleotide. So the nucleotide is made of 3 parts. Phosphate in orange, which is the same phosphate as chemistry, PO4, deoxyribose sugar, and finally a base. And these black lines, they're showing the bonds between each of these 3 components. Now, the base can be changed out. And we're going to see on the following page, it can be A, T, C, or G. These are what we call nitrogenous bases. So I'll show you more information of that after. Now, what I drew here, which is a really poor drawing, is that this only shows 1 nucleotide. Well, in humans, we have 3 billion nucleotides. So we have them essentially linked together in a chain. So this showing 1 single one will have many more attached to it. So that's what I've drawn in red here, which is a poor drawing, but, and so these will continue. So you'll have like many nucleotides connected together. And so what you end up with is this picture at the side, which is the DNA helix. So in the middle of the helix, thinking of it as like a round staircase or a round ladder, in the middle of the ladder, you have the bases, which are A, T, C, or G. The outside of the ladder, this whole component, we call it the DNA backbone. And that part is composed of the phosphate and the sugar. Okay, so we have sort of those 2 components sort of differently located. Now, we call this a DNA double helix because it has the two sides, okay? Something like RNA, that just has one strand. This has two strands which are across from each other. Okay, so I want to just zoom in a little bit on the bases. So again, that would be like this little green box that I've shown over here. These bases are universal. So that means that every living thing has these 5 bases essentially in their genetic material. And I say every living thing, it also includes viruses. So viruses also are made of these same bases. So one of the really strong supports for the theory of evolution is that, oh my gosh, all of these living things actually are composed of the same chemical components. So you'll notice that there's kind of like two different sections shown here, which are the purines and the pyrimidines, and they differ in their shapes. So the purines are going to have 2 rings attached to each other. Those are guanine and adenine. So we short form these typically to just A and G. I always remember those thinking of pure AG, which is pure silver. That reminds me that A and G are examples of purines. And the pyrimidines, you can see, are single rings. So the single rings are cytosine and thymine. Now over here, I've also listed uracil. That's also considered a nitrogenous base. Uracil is only in RNA, and thymine is only in DNA. Again, next year in grade 12, we talk about more details of RNA. For now, we don't really talk about it at all, actually, in this unit, but we really focus on the first 4 that are listed here. So the bases will always be across from each other. So I'm going to scroll back to my diagram here where I said, really, I have 2 strands. In the middle, I have nitrogenous bases, and on the outsides, I have those backbones. So in the middle here, the bases do not just randomly interact with each other. It's a very, very specific interaction, which is called complementary base pairing. So with the complementary base pairing, we will always have A, adenine, across from thymine. So a double ring across from a single ring. A and T, I always remember, it has 2 straight lines, which is like the A part and then the T part. They are always across from each other and they form a double bond, which is a hydrogen bond if you've taken chemistry before. C and G, which is cytosine and guanine, they are always across from each other. And they form a triple bond, which again is a triple hydrogen bond. If you've taken chemistry, you already know this, and if you haven't, that's okay. You'll learn it in grade 12 bio, but these interactions are extremely, extremely strong. And the reason why it has to be so is that the DNA contains information for the cell. As soon as those bonds, those hydrogen bonds are broken, the cell cannot read that information and therefore cannot actually make that instruction happen. So the importance of this concept of complementary base pairing, where I'm really showing it is across from each strand. So DNA replication, which you learn about in grade 12, it really, really relies upon this complementary base pairing. And really what it allows is to make sure that the DNA doesn't have errors in it. So whatever is on one strand, we can easily predict the second strand. The DNA replication machinery also checks and edits the information so as to make sure that there's no mistakes. So it basically is using the sequence to check for mistakes. So I put an example here. If strand 1 was G, T, C, C, A, T, etc., what would be across from it should be the complementary base pair. So across from the G, we should find C and so forth down the line. So again, I use the little trick of these two, A and T, have like a straight line across them. C and G are both rounded, so they will pair up together. And then I've shown actually what if there were an error. So accidentally, the wrong base was put into place. Now, this happens like 1 in every something like 1 million base pairs. So it's a very small amount. But as we know from grade 10, that can lead to some dangerous things. That is a mutation or a mistake, okay? So I've mentioned right here that the cell actually does check the DNA many times. In grade 10 science, when you talk about the cell cycle, you might have talked about cell checkpoints. And so it essentially is checking around and seeing if there's any mistakes. If there are mistakes, the cell will try to edit them, most of which it can do, but occasionally it cannot. And so the cell will enter what's called apoptosis. The cell will start to self destruct, basically to stop it from spreading to other new cells. So the last one I'm just going to mention here is the amount of information that DNA contains. So in one single cell, considering 46 chromosomes, if we were to stretch out the DNA strands, it would be over 2 meters in length, which is very, very long. So obviously those 2 meters have to be packaged down really, really small. So what I'm showing in this diagram is essentially how those nucleotides arranged in the double helix become coiled around proteins called histones. So histones are basically like, I think of it as the spool that thread comes on. So the little plastic spool, and then the thread is all wound around that. So basically condensing it. The same thing is happening in DNA. It becomes very, very tightly coiled and tightly packed until we have sort of what we know from, especially from mitosis, which is a condensed chromosome. So this is how DNA is stored. Now, let's say, for example, that the cell needs some information from here. It needs to actually look at a gene. Well, the cell actually has a whole way to actually send things in and undo the specific sections that it wants to look at. It will never, ever undo the whole thing at one time because it's 2 meters long. It will not fit in the cell. So I hope that this helps explain a few of the different parts of DNA. For tomorrow's lesson, there's not going to be a video. All you're going to be doing is playing around with the gizmos. So if you haven't done it yet, I would just make sure to start up your Explore Learning Gizmos account. The instructions to do that, you're going to find them over here on content. So I'll just show you really quickly on content and in course introduction. So you will need to create yourself an account if you didn't already. Okay, and then that should set you up really well for lesson number 2.